Science on the Menu, a podcast by the European Food Safety Authority.
Hello and welcome to another episode of Science on the Menu. My name is Ed Bray and I work in the communications team at EFSA. And joining me today is Jean-Loup Dorne, who is a senior scientific officer working on scientific methodologies in risk assessment. Welcome to the podcast, Jean-Loup.
Thank you very much, Edward.
Today we're going to be talking about animals, but not the animals that are on our farms that are producing food and drink. We're going to be talking about animals that we think about less in the food safety system, and that's the animals that are tested on to ensure that substances in the food system are safe. And we're going to talk about some more recent scientific developments that could help us to move beyond animal testing altogether. So first, Jean-Loup, what is the purpose actually of animal testing?
Historically, animal testing exists since the 1960s, after the famous thalidomide incident discovered in Australia.
Can you tell me more about that?
So thalidomide was taken as a drug, as an antidepressant or as a sleeping pill, and pregnant women took it, and then the consequence of thalidomide intake was basically children with no limbs. A medical doctor discovered that thalidomide itself was so-called teratogenic, so it basically blocked the development of limbs in the fetus because it went through the placental barrier. From that, the legislation was set to then test, very briefly, all those chemicals for their potential to have adverse effects, for example, on the fetus, but also on any organ of the body, be it liver, lung, and so on, to be very brief.
Okay, so essentially it became mandatory, is that right, to test on animals before certain substances could come to market?
It became mandatory, so it is set in stone within the legislation, put it that way.
Okay, and let's be clear, we're the European Food Safety Authority. We don't have labs at EFSA for animal testing. This is done elsewhere around the EU. Is that right? How does that work?
So depending if the compound is a regulated chemical, in the sense that industry submits the dossier to put this compound on the market, then we're talking about regulated chemicals such as food additives, feed additives, pesticides, and so on. Or if the chemical is an environmental contaminant, then of course, because it is a contaminant, it doesn't belong to any industry. So we would take for this contaminant the historical data we have, dioxins and so on, for example. When it is a compound that is about to reach the market, or an application for this compound to reach the market, let's take a fungicide or a food additive, the legislation requirements, of course, would ask for data on toxicity of that chemical on selected test species. We are talking about rat, mice, dog, and rabbit.
Okay. Often in toxicology, we talk about the dose makes the poison, correct? So I guess essentially that we are testing to see at what dose a substance becomes unsafe for firstly for animals, and then it's converted into something for humans. Is that more or less right?
Yeah, absolutely. So you have what we call dose response, which is Paracelsus, the dose makes the poison. From that dose response, we derive a no effect level, or if we do modeling, we derive what we call a benchmark dose limit. And from that, we apply uncertainty factors to convert the dose from animal data to human health, in the sense that there is what we so-called uncertainty factor of 100-fold, 10-fold to take into account the differences across species, and 10-fold to take into account the human viability from the baby to the elderly.
Okay.
That way.
Because basically humans are obviously different from animals, and therefore there needs to be a conversion factor essentially to convert it into something meaningful for humans. But I understand also a margin to ensure there's even greater safety. Is that correct?
Yes, absolutely.
We know that in society there's a lot of concern, rightly so, about the ethical value of testing on animals. And EFSA is doing what it can to move, in a direction where we don't need animal testing. And we adhere at EFSA to the principles of the 3 Rs. So that's to replace, reduce, and refine. Can you talk about this? The replace, I imagine, is to move to a system where we are not testing on the animals, reducing to reduce the number of experiments while getting the results we need, and refining to refine the experiments themselves to reduce suffering. Is that right?
Yeah, and on top of that, in the refinement, with the new methodologies that we've been working on for the last 4 decades, we can integrate some understanding of the processes that lead to toxicity, be it the toxicokinetics, which is what the body does to the chemical, such as metabolism and excretion, and what the chemical does to the body, which is the toxicity itself. Sometimes metabolism will bring something which is toxic, or sometimes metabolism will bring something which is not toxic anymore, and it's eliminated in the urine, for example. So using these approaches, we can combine historical data from in vivo testing, which is animal testing, but also we can look at cellular systems that allow you to look at a number of given organs that would be targeted by that chemical, be it the liver, the kidney, lung, what have you, and then combining that within algorithms which allow you to predict both dimensions, the fate, so the kinetics of the chemical, or the effect, the dynamics.
And that's where we are now. We're already moving in that direction, the replacing of the in vivo testing on the animal to other options. You talked about in vitro, in taking essentially cells of an organ and testing on those rather than the animal. But there is also new methods where we can do it with the data, the algorithms modeling of the data. And I understand that you've been working on a very specific model called TKPlate, which is moving us in the direction of reducing animal testing even further. Is that right?
Yes. So very briefly, TKPlate is an open access platform that allows you to model the kinetics of chemicals, what the body does to the chemical, but also the dynamics of the chemicals. And it's relevant to human health, animal health, but also to species of ecological relevance, so species that are in the environment.
Okay, so for example, a pesticide that is tested on to see what effects it might have on birds or insects or other species.
Yeah.
You say it's open source, so anyone could use it. Any scientist could come in, input the data at the beginning, choose the target organ, I guess, and then the outcome. Is that right?
Yeah. So of course, if you can register, and then you would have complete access to the platform. The first thing you would do is to select the model itself for a given species. And here we're talking about having models for humans, all the test species, be it the rat, the mice, the dog, the rabbit, and farm animals, pigs, cattle, sheep, and chicken, for example. And we are planning to put more models inside that. So you would choose your model, you would choose the chemical. So there is a link to a database, but you can actually put your own chemical if it's something for which we don't have so much data. And from there, you can actually select your exposure patterns. Let's put it in simple terms, the exposure to that chemical. And it could be an acute exposure, which is, for example, just one dose, or it could be a chronic exposure, which is a dose you would have every day. And from there, we could, for example, simulate in vivo studies without testing for 90 days, which is the standard, or for several years. And you can simulate, again, the kinetic of the chemical and go to the dose response to then calculate your safe level without testing on any animal.
Is this being used already, or does something need to be to change for us to use it systematically in risk assessment?
So when we published the database, we took quite some care to, first of all, produce a user guide, which is available to users, but also concrete case studies to take the user through the different steps on how we can really apply it to contexts of relevance, but let's say a food additive, a pesticides, and so on. And of course, you generate the data sets from the simulations. So we've taken quite some care to take the user through every step, all the way from inputting the data, choosing the model, all the way to downloading the data and generated what we have, which we call an automated report.
And I imagine the more data we gather, the more can be put into the tool, the more, the more established it becomes, and therefore the more useful it becomes.
Absolutely. And we gain experience. And of course, these models are so-called generic models because they can be applied to any chemical for a given species. However, these models evolve as we understand the biology of organisms because they get more refined.
Okay, which could also mean greater safety for us. We know more about the substances, and we're able, therefore, to understand the effects better as well.
Absolutely. So the consequence of metabolism can be the compound is eliminated
from our body.
From the body, or it can be bioactivated, which means that the consequence of metabolism is the compound becomes more toxic. So if we understand the difference between these two processes, we can actually determine if it's what we call the chemical itself, the parent compound, or the metabolite that will exert its toxicity in a given target organ, such as the liver.
And could you imagine this actually changing the whole process of risk assessment altogether, that in the future, if, as you said, that there were legislative changes that happen, that we could actually have a world without any animal testing at all? Could that be possible?
Well, I think it's already possible to reduce it, I would say, at least by a significant amount. You could do targeted testing by identifying, using, again, cell systems, even in animal cell systems and human cell systems. You could identify the targets, be it the liver, be it the thyroid, be it the lung, be it the kidney, and you could test that within human cell systems and, let's say, test species cell systems such as rats. Eventually, if there is a legislative need to get an in vivo study done, then you could, instead of having different studies, you could have a very targeted study, and it could be just one. So that would be a reduction already of 90% of the testing.
Wow. So it's potentially a very exciting development in how risk assessment, is done and will be done in the future. Well, I think that's all we have time for now. Thank you very much for joining us, Jean-Luc, and explaining about this exciting development in scientific methodology. Thanks to our listeners for joining us for another episode of Science on the Menu. We hope we'll see you again next time. Please subscribe to our social media channels where you can also find more material about our episodes. But for now, it's goodbye from me. It's goodbye from Jean-Luc.
Thank you so much, Edward.
And I hope to see you again soon on Science on the Menu. Bye.